Hello everybody. Welcome to the Antibody Society webcast series on antibody validation. In this, the 7th episode of the series, we're going to look at one of the most useful technologies for antibody validation, genetic knockdown. The Antibody Society is an international non-profit association. We're an international forum for facilitating discussion, disseminating information, providing guidance, and assisting in training and advancement of students and postdoctoral fellows. So if you care about antibodies, please join the Society. The speakers in this webcast series are experts from academia and industry, and they're expressing their independent assessments. They're not representatives of the Society. Viewers can write in questions at any time during the broadcast for the speakers to answer, and the speakers will remain online for 15 minutes after the cast has ended to answer questions. In this episode, I'm very happy to be able to welcome Dr. Travis Hardcastle from Horizon Discovery and Dr. Alejandra Solache, Vice President of New Product Development at Abcam. Travis will consider the various common gene suppression technologies, and Alejandra will tell us about their extensive practical usage in Abcam. Greetings, Travis. Greetings, Alejandra. Great that you can be with us today. We've seen in previous episodes that optimal commercial antibodies are both hard to find and to unequivocally identify, both at the point of sale and in the resulting literature. In any event, it's up to you, to the users, to verify that the antibodies you use are fit for purpose in your experiments, and this always needs adequate positive and negative controls. The question is, what are accessible forms of validation technology to control antibody specificity? You may be able to buy or make an appropriate recombinant protein for Western blot, ELISA, or IP validation, but of course that isn't always possible. But for specific cellular expression, or for expression in animal tissues, the options are more limited. One of the most useful technologies is to specifically ablate the expression of the target protein in the cell or tissue of interest. This is not a technology that every lab has mastered and takes much time and resource. Today, we welcome Travis Hardcastle of Horizon Discovery, who will describe their experiences in developing accessible cell genetic ablations. Welcome, Travis.
Hi, Simon. Thank you so much for inviting me to give this talk today at the Antibody Society, and thank you for having us on here and thinking of Horizon to talk about this subject. So what I really wanted to talk about today is cell line knockouts and knockdown technologies and how they can be used for antibody validation studies. So what I want to start off with is this 5 basic methods for validating your antibody. And I want to start off by saying that this has been adopted from a report that was in Nature Methods back in 2016, and where they really pointed out and highlighted these 5 basic methods. And so what are the 5 methods? One is orthogonal studies. Another one is independent antibody technology or techniques. Third is recombinant expression. And the fourth one would be migration capture, MS. And then the fifth and final one, which is one that we'll expand upon in a little bit more detail later on, is just genetic strategies for antibody validation. So the first technique that we're going to kind of take a 10,000-foot view of is this validation by orthogonal studies. And so what is this methodology and how is it performed? So basically you see with this technique, the antibody is validated by comparing the results with a non-antibody based method across multiple samples. And so as you can see in these 2 panels here, we have 2 different samples or 2 different ways of performing this technique. And so in the panel on the left, we have 2 different cell lines that have varying or drastically different expressions of this RAB27A protein. So in SKMEL30 cells, we have this very robust high expression, whereas in CaCO2 cells, we have a really low expression. And what you can see with the Western blot image in the panel is that the antibody really correlates to the protein expression based off of RNA-seq data. And what that does is it really further validates that antibody for that protein of interest. If you look at the panel on the right, the same technique can be performed when you use multiple tissue types. And so if we look at the expression of SLC2A2 in liver cells, you see that we have this really robust expression, whereas it's drastically different in the colon cells, where it's virtually non-expressed. And so what you see in the two images is that the antibody staining for SLC2A2 in these two different tissue types really represents the RNA-seq data, and it further validates the specificity of that antibody for SLC2A2. Another method for validating antibodies is using the validation by independent antibodies technique. And so for this technique, antibody specificity is demonstrated by comparing two antibodies targeting different regions of the same protein. And so what we have here in these two panels is, in panel A and panel B, is two anti-TCHHL1 antibodies, and they're staining across several different tissue types, such as cerebral cortex, kidney, lymph node, and skin. And as you can see, that the staining across all these different tissue types is relatively the same for the two different antibodies that are targeting different regions of the same protein. And so what that does is validate both those antibodies for being highly specific for the TCHHL1 protein, and it validates the antibody. Another technique for validating antibodies is to use this validation by recombinant expression. So in this technique, the antibody binding is verified by using an overexpression or an overexpressed protein or a tagged version of the targeted protein. So for example, in these images, we have this anti-nestin protein, which is highlighted in green here in the center panel, and what you also have is this GFP tagged nestin protein in the left panel, which is represented by the purple color. And as you see, when you overlap the two images, you almost have 100% overlap between the green and the purple, which, as we've said before, really validates the specificity of the antibody for that protein because of the overlap between the GFP tagged image. One final technique that we'll look at it from a high view is this validation by migration capture MS. And so for this technique, the staining pattern and the protein size detected by the antibody is compared with results obtained by capture mass spectrometry. But this technique does have its limitations, and one of those limitations is that when the target protein cannot be detected by mass spectrometry, you're not getting the readout you're looking for, and so therefore it's a huge limitation. So on to what we really want to focus on today, which is this validation by genetic studies or genetic strategies. For this technique, the antibody specificity is confirmed by downregulating or knocking out the target protein on the genetic level by using either siRNA or an RNAi technique or CRISPR-Cas9. And so what we have here in this image is a representation of that. And so we have three different lanes here, and the first lane is a siRNA that has a control or a scramble sequence in it and that doesn't target anything within the genome. And then you have the two other lanes that correspond with two different siRNAs targeting the protein of interest, which in this case is PPIB. And as you can see, in both the lanes that have the siRNA targeting PPIB, we have almost no banding pattern for PPIB, which really in two different samples shows the specificity of PPIB for the antibody for PPIB, specificity of it for these cells. So if we focus on the two tools that are commonly used to decrease or knock out gene expression, you see that the two main technologies are RNAi, which exists in the form of siRNA, shRNA, or microRNA. And what I'm really trying to get out of the slide is that this technique and this technology really acts on the mRNA level. And so what that means is that with RNAi, it's not a permanent change. And so when you knock down a protein using RNAi, or such as siRNA, you get this temporary downregulation of the protein of interest, but over time, the protein will be rescued back up to normal expression levels as the siRNA degrades, and you get this natural turnover of protein and expression of protein. The other technique that's used to decrease gene expression or completely knock out gene expression would be using a gene editing technique, and commonly that's performed with the CRISPR-Cas9 these days. One of the major themes of that is that this happens at the genomic level. So what happens is you have a genomic alteration that creates a knockout or an ablated protein, and what you have is that gene gets completely turned off. And one of the key benefits of this is that this expression is turned off and it's heritable over time. So as the cell divides, that same mutation will be passed down from cell to cell, and that ablation of that protein of interest will be carried out over time. So if we look at some of the advantages of siRNA compared to another technique, you'll see that one of the major advantages of using siRNA is that it's fast. And what that means is that these knockdown experiments can be performed in a relatively quick manner of 72 to 96 hours. And so what that means for researchers who want to validate their antibodies is that in as little as 3 to 4 days, you can validate that antibody, your antibody of interest for a particular protein using an siRNA method or technology. Another, I think, advantage of using siRNA is that it's very robust and it can be used across multiple cell line backgrounds. And so what that means is some cell lines, as we saw earlier, express proteins at different levels. And what you do is you knock that protein down in different backgrounds and really then based off the antibody staining, it really validates your antibody over multiple cell line backgrounds. Another thing that can be used with siRNA is like we said earlier, it's not heritable, and it'll be, the protein expression will be rescued over time. Is that using siRNA, you can really test the sensitivity of antibodies because of the fact that you will get this initial decrease in 72 hours, but then over time that protein will be rescued and will come back to normal states, and you can see exactly how sensitive your antibody is for the protein by seeing it at its lowest levels and as that protein begins to be re-expressed over time. So one of the other techniques that we talked about was using a knockout cell line. And so here at Horizon, one of the main technologies used to generate our knockout cell lines is the CRISPR-Cas9 system. And so what that is at a 10,000 foot view is it was just a mechanism of adaptive immunity that was developed in bacteria. And what it was is it evolved over time to adapt and defend against foreign genetic transfer bacteria in the form of phage or horizontal gene transfer. And what we've done and been able to do, technology's been able to do is over time, is we've really been able to harness the system and use that in mammalian cells and create these genetic knockouts or these gene mutations in any cell line that we can grow in the lab. And so what are the two requirements for the CRISPR-Cas system? Well, one of the requirements is a Cas9 nuclease. And what this is it's just an enzyme that when guided to DNA will act as molecular scissors and create these double-stranded breaks in the genome. So what is the other component? I think it's somewhat maybe obvious, but it's the guide RNA. So we need some sort of mechanism to guide this CRISPR-Cas9 nuclease to the genomic location of interest. And so what you have is you have to have a guide RNA to do that. And so what we have here in this diagram on the right is just a basic workflow of how this technology is used. And so what you do is you transfect your cells with both of these components. And so for the Cas9 component, that can exist in either the form of Cas9 protein, Cas9 mRNA, or even in the form of plasmid, which isn't shown on here. And you co-transfect that with the guide RNA, which can exist in this diagram. It exists in the two-component system, but this can also exist in a single guide component in which you have an extra stem loop over here, which makes it one whole sequence as opposed to being two components. And so what happens is from 48 to 72 hours after transfection, the Cas9 will be directed and guided to the genomic location of interest by the guide RNA, in which you have this 20 base pair complementary binding. And whenever that binding happens, you have a conformational change and you create a double-stranded break for the Cas9 nuclease. From that point, we take advantage of the cellular's repair mechanisms, in which aren't always perfect. And so what oftentimes happens is that you get a repair that is not optimal. And so for some cells, you will have a 1 base pair insertion. For other cells, you might have a 4 base pair deletion. And what happens is researchers can then, at that point, after this initial 72 hours, is dilute their cells out, their population of cells out, and then form single cell colonies in a 96-well plate and allow the cell line to grow out. From that point, after you get a robust enough population, researchers can design simple primers that are on the outside of where this genomic break should have happened using the Cas9 system, and use Sanger sequencing to further characterize what the indel of interest is for that particular clonal cell line. And so we've really harnessed this technology at Horizon Discovery and have been able to create knockout cell lines, a whole plethora of knockout cell lines. But first, I want to just highlight what are some of the advantages of using a knockout cell line for antibody validation. And as I alluded to earlier, that knockout cell lines, at least from Horizon's perspective, are all validated at the genetic level. And so we perform Sanger sequencing on all of our cell lines in-house, and that really allows for us to characterize our cell lines. And what you get is this functional protein expression that is ablated, by either the introduction of a frameshift mutation or just a deletion of a large sequence of this genomic DNA as well. And so what we have here in the panel on the right is we have images of two different cell lines. One is our wild type cell line, which expresses all of the protein of interest, which is SLC30AC in this instance. And as you can see, we have a blue stain representing the nuclease, a red stain that represents the microtubules, and then this green immunofluorescence that represents this SLC30A6 protein in our wild type cells. And so what you have on the panel on the right is our knockout cell line for SLC30A6. And as you can see, that protein has been ablated, and it's really represented by the antibody that was chosen for this particular experiment, which further validates that antibody as being specific for just that protein alone. And so I just want to say that I think this really points out the validity and the robustness of this technique in using knockout cell lines for validating your antibodies and specificity of your antibodies. And I want to say that there's these many knockout cell lines have already been used to validate antibodies by many commercial suppliers, as Simon alluded to in his introduction. And I will say that back in 2015, we had an agreement with a commercial supplier which they use our cell lines to validate their antibodies with. And I'm happy to say that as of the time of this webcast, we will have signed an agreement with KTH Rolla Institute of Technology and the Protein Atlas for them to use our knockout cell lines in-house to validate a lot of their antibodies of interest. So I think it really goes to show that it highlights how great this technology is and how great how Horizon's been a trusted brand to provide these sort of knockout technologies for clients and performing these validation studies. I just want to close up with one slide real quick. I wouldn't be a good product manager if I didn't highlight what we have in-house and what Horizon has in-house and our ready-to-go knockout cell lines. I elaborated on how you would perform these knockouts and how you would create these knockout cell lines, and I made it seem somewhat trivial, but it's not as easy as I made it seem, and we've really been able to harness that technology, and we have a robust and very large portfolio of cell lines for anybody who's looking to validate their antibodies in-house and doesn't want to go through the process of generating a knockout cell line. So we often generate multiple clones for the same protein of interest, and we pair these knockouts as a standard. We pair them with a match control to have the side-by-side comparison, just to provide the further confidence that the antibodies that are tested are completely specific for the protein of interest. And so in-house, we can target about 9,000 genes. We have about 3,000 of these right now that are available to ship off the shelf. And as I said, we have multiple clones for each of these cell lines, which means we have well over 7,500 off-the-shelf clonal cell lines that can be used for researchers in validating their antibodies. We also do have the capabilities to perform custom projects for customers if they should know where the epitope the specific epitope's at, where we completely excise that for an antibody, and so it gives you even further confidence that your antibody is very specific and validated in it that way. In the panel on the right here, I just want to highlight that this is just 6 of the cell lines that we have in-house and their corresponding antibodies for those proteins of interest. And as you can see, you can really validate the antibodies by the complete ablation of the protein, as you can see across this panel of cell lines that we have. And so I think with that, I will say thank you for listening, and I will be open to answering any questions you may have.
Thank you very much indeed, Travis. That's a very, very nice presentation, and it's obviously good. Firstly, that Horizon has helped with the webcast series and presenting today, and that you have a bank of knockout cell lines so that users can directly obtain a negative control without weeks or months of work, and I think that's extremely useful for the community. I wondered, are your cell lines, I guess they're predominantly human?
Yes, Simon, that's correct. We do have many of the common human cell lines that are used at least for cancer research in-house. So HCT116, RKO cell lines, et cetera. But I think our main technology and one of the main cell lines that we use that allows us to do this is our haploid cell lines, which is a near haploid cell line. It is a derivative of the KBM7 cell lines, and it only has one allele for most of the genes in the genome. And so what that really does is it allows us to really target that protein that we want without having to worry about compensation by a second allele or knocking out that second allele. And so it's really beneficial technology.
That's super. Thank you very much indeed, Travis. We've seen that cellular knockdowns and knockouts are valuable for antibody validation, but how are they to use in large-scale practice? Some 4 years ago, the global life science company Abcam started to use knockdown cell lines for antibody validation on a large scale. So it's a pleasure to be able to welcome Dr. Alejandra Solache, Vice President of New Product Development at Abcam, to tell us about their experiences using such technologies. Welcome, Alejandra. Welcome to the series. Would you like to tell us about what your company has done using knockdown technologies?
Yes, absolutely. Thanks, Simon. And thanks to the Antibody Society for inviting us to participate in this webinar series, as I believe that this is a topic that is key to our scientific reproducibility. And today I will discuss how recombinant antibody technologies, as well as high standards in antibody characterization in multiple applications, particularly utilizing knockout technologies and other validation strategies, can support scientific reproducibility. So generating high quality antibodies requires effective strategies for immunogen design, protein expression, antibody discovery and manufacturing, as well as specificity validation and strict batch testing. The choice of the antibody format combined with high standards in manufacturing has a great impact on the specificity, antibody performance, and consistency. At Abcam, we are utilizing best-in-class antibody discovery platforms to develop the highest performance antibodies and focusing our efforts on generating recombinant rabbit monoclonal antibodies through standard platform for rapid rabbit monoclonal technologies through hybridoma conversion, B-cell cloning, NGS, and phage display. And the result of these features is recombinant rabbit monoclonal antibodies that are specific and very sensitive to the target molecule of interest. When compared to other common antibody formats, such as polyclonal and standard hybridoma-produced antibodies, recombinant rabbit monoclonal antibodies have multiple advantages, including improved consistency and reproducibility, as well as the highest affinity and specificity. Therefore, in terms of antibody selection for your own experiments, a recombinant antibody is the most robust format available, and we highly recommend them if you have a choice over a standard hybridoma monoclonals and certainly over a polyclonal. However, regardless of the antibody format, antibodies require in-depth characterization in order to ensure that they only bind to the target of interest in multiple applications. At Abcam, we are working towards providing the highest level of characterization and validation and striving to get the highest possible batch-to-batch consistency. And today I will discuss how Abcam's validation is constantly evolving by adopting new technologies to assess specificity. In particular, I will share with you one of our most exciting initiatives, which is the incorporation of knockout technologies in our antibody validation. So we have incorporated a range of assays and applications to ensure that we can assess specificity and selectivity during early development of these rabbit monoclonals. And at the clone screening, as well as the final characterization of the antibody. And we aim to provide validation in many applications as many applications as possible, including Western blot, IHC, ICC, and flow cytometry, among others. But we select the most appropriate application based on the particular protein targeted, and we use a combination of approaches to confirm the specificity of all of these antibodies. And we strive to select the screening material to match as closely as possible to the samples that you will utilize in your own experiments. In this particular case, for example, I'm showing an example where IHC is the application of choice, and we are utilizing multiple tissues and multiple species to assess the specificity of the antibody. And we also extend our validation to the relevant tumor tissue when this is necessary. And as you can see here, this is the case of an anti-PD-L1 antibody where we are testing in multiple tumor samples in a tissue microarray, and also including the normal control tissue. And this is very important, as well as utilizing the right positive controls. We always try to identify a negative control in which the target protein of interest is not expressed. But for some particular targets, finding a negative control is quite challenging. And this is one of the reasons why knockout models are key in the validation arsenal. So we have taken these knockout models as a standard, one of the highest standards for antibody validation, and as they represent true negative controls for most of the proteins expressed. And for the last 4 years, we have implemented a large-scale antibody validation using knockout models. In this particular case, for example, we have an anti-PKC antibody, PKC alpha antibody, where we are showing that in the wild type, the protein of it, the band is showing at the right molecular weight. This band of interest disappears within the knockout, and then we also include some tissues or cell lines where the PKC alpha will be endogenously expressed as controls. And this is what you will want to see for an antibody to demonstrate the specificity, to confirm the specificity of this antibody. The flip side is when the protein of interest is actually not expressed in the knockout, and you still see a band in the size where that particular protein should be expressed, as it is the case in this blot on the right where we are using anti-ILK. And in this particular case, this is telling us that this particular antibody is detecting some off-target, is having some off-target binding, and is not fit for purpose, and therefore is being removed from the catalog. So we have taken a thorough approach to validating multiple antibodies to a particular target, because it is very important for us that you have choice for your own experiments, and we will want to have multiple targets, multiple antibodies to a particular target that have been confirmed specificity. In this particular case, we have 5 antibodies to annexin-1, and this technique is really allowing us to confidently identify the antibodies that have specificity, as well as the antibodies that have off-target binding, as it is the case in here with a rabbit polyclonal against annexin-5, which actually show off-binding effects, and it has now been removed from the catalog. And as you will see, this is what you will see. This product is no longer available in the catalog. So importantly, as I mentioned before, this effort has been done above and beyond to the already comprehensive validation that we use from the beginning of since we started. So, but it's been a really great initiative for us. We know that not all of the antibodies will work in Western blot. Some antibodies will work, but are more suitable for flow cytometry, ICC, or IHC. And we have tried to test these antibodies in the knockout models in the particular application where it's more relevant for them. In this particular case, we are showing flow cytometry with the wild type and knockout, as well as ICC wild type and knockout, as I had already shown Western blot. But obviously doing this validation in multiple applications and with multiple antibodies is a really large effort. And this just wanted to give you an idea of the scale of this initiative. We have so far done about 90 knockout flow cytometry tests, to over 200 ICC tests, over 7,000 Western blots. And with this initiative, we now have confirmed the specificity of over 2,500 antibodies. And actually, we have been able to remove the non-specific antibodies. And we have now covered over 2,000 protein targets. And importantly, we have learned a lot through this initiative. We have found that that recombinant monoclonal antibodies are consistently showing to be the highest rate of specificity to the targets compared to traditionally produced monoclonals or polyclonal antibodies. And as you know, polyclonals have inherent batch-to-batch variability, and each batch needs to be checked. And what you can see here is that while the polyclonal performs well and it has specificity for the target protein, some batches actually show off-target binding, which is extra bands that are not eliminated in the knockout. And in contrast, we have found that the recombinant rabbit monoclonals, the batch-to-batch variability is virtually eliminated. And when confirming specificity of all these batches, we see pretty much all of the batches perform the same. Another important learning is we have found during the course of this initiative is that a significant number of top-sided antibodies show off-target effects. So when choosing antibodies for your own research, go for those that have been confirmed specificity, in multiple applications, and ideally that have been validated with knockout models. If the antibody you need to use has not been knockout validated, then I would recommend that you validate them themselves, even if they have been previously cited. So confirming the specificity of an antibody takes effort. We definitely know that, and it requires the right reagents. So therefore, we are making the relevant knockout cell lines available for people to utilize them themselves on their own labs, for you to utilize them in your own labs. As we know that it takes a lot of time to generate these knockouts, up to 16 weeks, and they require validation too. So we are making this range of cell lines available off the shelves and validated and ready to use. So in summary, I have hope that I have shown you how we are utilizing knockout validation for our antibodies, providing products which are specific and work the first time is integral for our mission. And this is particularly important for complex assays such as multiplex IHC, where the objective is to accurately detect the presence of multiple proteins in a particular sample simultaneously. And in this image, you can see the presence of 6 individual proteins clearly identified. So I hope I have given you a flavor of how our knockout cell line initiative and other validation technologies are enabling us to confirm the specificity of our antibodies and how this is having a positive impact on the quality of experimental data. And with that, I'll be happy to take any questions.
Thank you, Alejandra. I think many users will be overwhelmed by the diversity of potential validation technologies, and you've led a large validation effort. So how would you assess your experiences with the value and reliability of data that one can get using knockout technologies in comparison to the other available methods?
Yes, so we really have chosen these knockout technologies because we feel that this is at a time in science where as science evolves, this is one of the best techniques available for us and for scientists. With the new knockout technologies becoming widely spread, it is really one of the best technologies. For example, compared to knockdown, where knockdown is a lot more unreliable, and compared to other more complex types of applications such as mass spec and other applications that not everybody has access to. We felt knockout validation gave us the high level of a high level of stringency that we need to confirm specificity, as well as the ability to utilize it at large scale. Because we needed to go through this, we wanted to go through this as a large effort and not just be able to do it for a small number of antibodies. And we believe this is something that now, with the lines becoming available, people in their own labs will be able to do it much more easily than before.
Yes, thank you. I think that's very, very valuable. Thank you. So we've seen that gene knockouts in cell lines are a powerful tool for validating antibodies. They're versatile because they can be used in many common cell biological techniques and proteomic applications. And of course, now we've seen that many validated knockdown cell lines and lysates are commercially available. So in the next episode of the webcast series, we will look at some less conventional methods for validating antibodies, protein arrays, and IP mass spectrometry. This is the Antibody Society webcast series on antibody validation. Thanks for joining us, and bye-bye until next time. The series would have been impossible without the support of our corporate sponsors, to whom we're very grateful. Travis Hardcastle and Alejandra Solache will be online for the next 15 minutes and will be happy to answer any questions you might have. If you have any questions on the use of knockdown technologies for validating antibodies for Travis and Alejandra, please type them in at the question and answers tab.